Epithelial tissues are present in the human body on the outer covering, for example, the skin. Your skin is made up of epithelial tissue, and the linings of internal hollow organs, for example, the lining of the windpipe, the lining of the food pipe. All of these are epithelial tissue. The glands, glandular tissue is also epithelial tissue. What are the functions of epithelial tissue? Think of what your skin is doing for you. It is the outermost covering, so it is providing protection. Protection from all kinds of external hazards, protection from abrasion, from drying, from any kind of injury. Skin also controls exchange with the outer environment, how much the skin is going to secrete, okay? How much will the skin absorb? Filtration. Sensation is also a function of the skin. When you touch something hot, you immediately move your hand away. So it was the sensors in the skin that detected that you were burning your skin. The secretions, how much the sweat glands which are present in the skin, how much of the sweat would they be secreting? Sweat helps in cooling the body temperature. Epithelial tissues have several important characteristics or features. The first one is cellularity. That means the epithelial tissue consists of several layers of cells. It is made up of largely cells, and there is absolutely no intercellular space. So there is very little extracellular matrix, and whatever matrix is present is located in the basal foundation of the epithelial tissue, which is known as basal lamina. So if you look at the histological section of epithelial tissue, it would look like a brick wall where every brick is a cell. The second feature is polarity. Polarity means when there is structural and functional difference between the two ends or the two surfaces of the cell. So epithelial tissues have one surface that is unattached or free, that is known as apical surface, and the other surface is attached onto the foundation. The foundation is the basal lamina, and that surface is the basal surface. And these two surfaces are actually very different from each other, both structurally and functionally. This figure shows the polarity of epithelial tissue. You can see that there is an apical surface on which you might see microvilli or cilia, while the basal surface is fixed or attached. Continuing with the characteristics of epithelial tissues, attachment. The epithelial cells are attached on one surface, that is their basal surface, to the basal lamina or the basement membrane, which is the foundation. Avascularity. This is very important feature. Epithelial tissues do not have blood vessels, and that's why if there's a superficial scrape on your skin, you do not bleed. In order for a person to bleed, the injury has to reach the connective tissue layer which lies under the epithelial tissue. Arranged in sheets. The epithelial tissues have layers of cells arranged one after another, so they are arranged in sheets. Regeneration. Epithelial tissue has a really high rate of mitosis. The cells are undergoing rapid cell division. We are constantly losing epithelial cells, that means cells from the skin. The skin is shedding cells, and there is constantly new cells being produced at the base, and these cells are moving upwards. Epithelial tissue shows certain types of specializations. The first one is a microvilli. The microvilli are short finger-like projections of the plasma membrane of the cell, and the main function is to increase the surface area, and you can find microvilli in the inner lining of the digestive tract as well as the urinary tract. The second one is a stereocilia, which is a longer version of the microvilli, and it is found in specific locations such as the inner ear or the male reproductive tract. And the third one is known as the cilia. We usually have ciliated epithelium in certain locations, for example in the respiratory tract. And the cilia are short hair-like structures, and they show wave-like motions. And then using this motion, they move substances. For example, when you're breathing in air, along with the air, you are also inhaling dust and debris. So those dust and debris are captured by the mucus, which is present in the respiratory tract. And that mucus, along with the dust, is then finally pushed outwards because of the cilia motion. This figure shows microvilli and cilia on epithelial cells. The epithelial cells are connected on the basal surface to the underlying basement membrane or the basal lamina, And the basal lamina is made up of two layers. There is the upper clear layer and the lower dense layer. Under the basement membrane would be the connective tissue. Now, since epithelial tissue does not have any blood vessels, it is avascular. That means all the nutrition has to reach the epithelial tissue by diffusion from the connective tissue. And because the epithelial tissues are the outermost layer or the innermost layer of the hollow organs, they are exposed to abrasion and toxic exposure, chemicals. So these cells of the epithelial tissue need to be replaced constantly, and that's why a high rate of mitosis is required here. The figure shows the basement membrane with the clear layer and the dense layer and the connective tissue underlying the basement membrane. Classification of epithelial tissue. There are two ways in which epithelial tissue can be classified. The first is based on the number of cell layers. So epithelial tissue can have a single layer of cells, then that particular epithelial tissue would be known as a simple epithelial tissue. While epithelial tissue could also have multiple layers, that means more than one layer of epithelial cells, and that epithelial tissue would be a stratified epithelial tissue. Usually when there is more than one layer of cells, those are in areas where there is a lot of mechanical or chemical stress involved. While when there is a single layer, it is usually in places where you need efficient secretion or absorption. So simple and stratified epithelial tissues, that means single layered and multiple layered epithelial tissues, are shown in this figure. You can see in the simple epithelial tissue, the same layer has an apical and a basal surface, while in the stratified, because there are multiple layers, only the topmost layer would have the apical surface, which is the free surface, and the bottommost layer would have the basal surface that is attached to the basement membrane. The second basis for epithelial tissue classification is the shape of the cells. So epithelial tissue, if they are made up of flat or squished cells, those are known as squamous cells. So that tissue would be squamous epithelial tissue. If the epithelial cells are cube shaped, that means they have the same height as the width, then that is cuboidal cells. If the cells are taller than their width, that means they look like columns, that would be columnar cells. While there is a special class of epithelial tissues where the cells change shape, and that is known as transitional epithelial tissue. So this figure shows the three primary shapes: the flat squamous, the cube-shaped cuboidal, and the tall columnar. So for epithelial tissues, anatomists like to combine the two criteria for the classification of epithelial tissues. That means they want to describe any epithelial tissue based on its shape as well as the number of cell layers. So from this slide on, we will be describing each type of epithelial tissue, and you will see that there are two adjectives for that epithelial tissue. One describes the shape and other describes the number of layers. The first one is simple squamous epithelium. That means this tissue has this tissue is simple, it's one layered, and the shape is squamous. The location of this type of tissue is usually in the heart, in the blood vessels. What is the function? It reduces friction. An important example for location of simple squamous epithelium is the alveoli of the lungs. Stratified squamous epithelium. This epithelial tissue is flat, but it has multiple layers. Location is the lining of the mouth, of the anus, vagina. Function is mainly protection. This figure shows the stratified squamous epithelium. Now if you look at the figure, whenever there are multiple layers of cells in epithelial tissue, you won't be able to tell the shape of the cells if you look at the layers which are in the middle or at the basal surface. You always have to look at the apical surface layer. That means the topmost layer of cells. That layer of cells gives you the information on the shape of the cells. That means whether the layer is squamous or cuboidal or columnar. Simple cuboidal epithelium, that means epithelial cells which are single layered and the shape is cube shaped. Location is in the thyroid gland or in the tubules of the kidney. Function is secretion. Important example for location of simple cuboidal epithelium is kidney tubules. Stratified cuboidal epithelium, that means multiple layers of cube-shaped epithelial tissue. This is a rare tissue. Location is in the ducts of sweat glands. Function is secretion or absorption. Important example, sweat gland duct. Simple columnar epithelium, that means one layer of tall column-shaped epithelial cells. Important example for location is lining of stomach, intestine, etc. Function is secretion, absorption. This figure shows the simple columnar epithelium of the intestinal lining. Stratified columnar epithelium, that means multiple layers of tall column-shaped epithelial cells. Important location is pharynx, mammary glands. Function is protection. The figure shows stratified columnar epithelium in salivary gland ducts. This is a special category of the epithelial tissues. It's known as pseudo-stratified ciliated columnar epithelium. The word pseudo means false or fake. So this is a fakely stratified. That means it is not really stratified, but it gives you the false impression of being stratified. That's because the nuclei are situated at different levels. Usually in a simple layer, that means in one layer, the nuclei would be all at the same level. And when you see nuclei at different levels, that's when you think that the tissue is stratified. So it is giving you that false impression. It is ciliated, that means it has cilia on it, and the cells are tall. Location, please remember trachea. That's a very important example. What's the function? It's protection and secretion. So this figure shows you the pseudostratified ciliated columnar epithelium in the trachea. You can see the cilia or the small hair-like structures on the apical surface. Look how the cells near the base they don't give you the shape of the cells. It's only the cells near the apex that give you the columnar shape. Transitional epithelium is also a special category. The name transitional means that the shape of the cells keeps changing. So an important example for location is the urinary bladder. Now imagine the urinary bladder. It can hold urine and it stretches as the amount of urine inside it increases. So the shape of the cells depends upon the amount of stretching. This figure shows you how the transitional epithelial cells change their shape. There are two panels. In one panel you have a bladder which is relaxed, and in the bottom one the bladder is stretched because of the accumulation of urine, and in the corresponding histological sections which shows the cells, you can see how the shape of the cells are changing from the relaxed epithelium to the stretched epithelium. Epithelial tissue also makes up glands. What is a gland? A gland is made up of one or more cells that produces a secretion. And what exactly is a secretion? A secretion contains protein molecules in a watery medium, that means an aqueous medium. So there are two main types of glands based on their mode of secretion in human body. These are endocrine glands and exocrine glands. An example of exocrine gland is sweat gland or the oil glands on your skin. So the exocrine glands, they make the secretions, and then the secretion goes out from the inside of the gland through a duct or a tube. While the endocrine gland, they are known as ductless glands because they do not have ducts. So whatever secretion is synthesized inside the gland, it just exocytoses out. That means there is exocytosis that takes the secretion out, and the secretion goes into the surrounding medium, which is usually the interstitial fluid or blood. And eventually all of the secretions from the endocrine glands reach blood, and then blood takes it to its destination. And the secretions coming out from endocrine glands are hormones. Exocrine glands are of three types: serous gland, mucous glands, and mixed exocrine glands. Serous glands produce a watery secretion. Mucous glands produce a more viscous secretion because there is glycoprotein in water which makes mucus. And then the third type is the mixed exocrine gland, which contains both serous and mucus secretions. Endocrine glands, on the other hand, they do not have any other categories. All of the secretions are hormones and they come out by exocytosis. As mentioned in one of the previous slides, glands can be made up of one cell or more than one cell. If they are made up of only one cell, they are known as unicellular glands, for example, goblet cells. If they are made up of more than one cell, then they are multicellular glands and they also secrete mucins. Now, the multicellular glands can have different shapes. If they look like tubes, then they are known as tubular glands. If they have blind pockets at the ends, then those are known as alveolar or acinar glands. Sometimes the multicellular glands can have a tube-like structure which ends in a pocket-like structure. Then it becomes a tubulo-alveolar or a tubular acinar gland. Multicellular glands can further be classified based on whether their ducts are branched or not. If you look at this figure, there is some part in blue which is the duct and the pink part is the secretory cells. So if the blue part, that means the duct, is branched, then it is not a simple gland. A simple gland would not have branching ducts. And simple glands can also have different shapes. They can have a tubular shape, they can have an acinar shape, and so on. If the multicellular glands show a branched duct, then it is known as a compound gland. For example, the pancreas. And the compound glands can also have different shapes. They can have tubular shapes or acinar shapes. Glandular epithelium has three mechanisms of secretion. The first one is called merocrine or eccrine secretion. Merocrine secretion is simple exocytosis. If you look at the figure, the middle panel, it shows that there are the purple shaped cells, the blue colored nucleus and the dark purple Golgi apparatus. A secretory vesicle comes out from the Golgi apparatus. The secretory vesicle starts moving towards the cell membrane. It fuses with the cell membrane and the contents of the secretory vesicle are exocytosed out. An example of merocrine secretion is found in the salivary glands. The second mode of secretion is known as apocrine secretion. Apocrine secretion is commonly seen in mammary glands. In this type of secretion, it's only the apical part, that means the top part or the free surface of the cell, is secreted. Here you will see that the Golgi apparatus, it releases the secretory vesicles. All the secretory vesicles, they start accumulating in the apical part, and then the entire apical part along with the cytoplasm, it comes out or is pinched off from the cell, and then this releases the secretory product. The third type of secretion is holocrine secretion found in sebaceous glands in the skin, and in this type of secretion, the entire cell bursts apart. So the secretory vesicles accumulate all over the cell, and then the whole cell, that's why the name holocrine, try to remember this, the entire cell would burst apart and the cells would be replaced by cell division from stem cells at the basal surface.